This is going to be AP Bio Unit 1. This is going to be covering the chemistry of carbon and functional groups. So the chemistry of carbon, when we talk about organic chemistry is working with compounds of carbon, which can covalently bond. I can have single bonds, triple bonds, double bonds. All of these different bonds are what kind of make organic chemistry organic, is because of the carbons. Organic compounds are going to be ones that contain carbon and hydrogen. If you ever heard of hydrocarbons, they're molecules that are carbon and hydrogen together. When I look at the periodic table, I can see that carbon is in group 4. Remember, when I take group 14, I can remove that 1 and I have 4 valence electrons. So carbon has 4 valence electrons. The interesting thing about carbon is for it to be able to hit that octet rule, I need 8 valence electrons. It has 4 already, and it needs 4 more to make 8. So carbon can bring in 4. So now I have a full outer shell. I have my octet, my valence electrons of 8. But another thing carbon can do is it can also get rid of 4. It takes as much energy to bring in 4 as it does to get rid of 4 valence electrons. So carbon can get rid of 4. So now I can either have a full shell or I can have an empty shell for carbon. This allows it to be able to bond real quickly with other types of molecules. Here I have some carbon bonds. Carbon can form again single, which are these 1 lines. Each line represents an electron getting passed back and forth. So if it's 1 line, 1 electron's getting passed back and forth. Here I have a double bond. These are 2 electrons that are being passed back and forth between these 2 carbons. I have 1 electron passed back and forth here, 1 here, but then 2 get passed between those 2 carbons. Here I have 3, so 3 electrons get passed back and forth these 2 carbons, and then 1 electron gets passed back and forth here. Carbon's going to use its valence electrons to form long chains, so I can make really long chains. I can branch off, I can make rings. So here I have a triple bond between these 2 carbons, singles here, I have single bonds here, triple again, I'm branched off on both sides. Here I have a ring. This dashed line is saying that I could possibly have 2 here, or I could have 2 here. If I had 2 being passed back and forth here, then I would have to have 1 here. So this is kind of saying that I could possibly have one, the dashed lines are a possibility. So the backbone or the skeleton of the carbon molecules, this is what's going to make most of these organic molecules. And they can be different shapes. So we call these ones that are straight linear. We have branched. These carbons are going to branch off other carbons. And then I can have these really ornate molecules start to form. I can have double bonded. I can have a ring, which makes a circle. And I can have these super long chains. We see these in lipids. And these are your hydrocarbons. These are all just basically carbons and hydrogens together. This is very nonpolar, which means it's not going to blend well with water. So we need to talk about functional groups. There are 7 main functional groups that we need to discuss. We're going to talk about hydroxyls, carboxyl, phosphates, aminos, carbonyls, methyl, and your sulfhydryl groups. And they all have different characteristics, so we kind of need to know what's going on with those. So let's look at hydroxyls. These are going to be considered polar. Polar means that it's going to dissolve in water. So here I have an R group. When you have an R group is saying that I have a whole organic molecule over here. I might have a carbon chain. I have other carbons and hydrogens and other things over here. And off that, I have a branch of an oxygen and a hydrogen. Sometimes you'll see it with a dash. Sometimes you'll see OH together, and that's showing that that's a hydroxyl functional group there on it. So here I have ethane. Ethane is a nonpolar. I have just carbons and hydrogens. This means that these are going to have a bond that's going to move back and forth. the electrical negative charge is very similar, so these get shared evenly. But, so we call it nonpolar, hydrophobic, won't dissolve in water. But when I look at this other molecule, I've added an oxygen. So all I've done is went from an H to an OH. This has now changed it into ethanol, which is now a type of alcohol. That OH now is going to make it polar, which means it's hydrophilic, it's water loving, and it's going to dissolve in water. So the question is, well, how does that make it polar? So here I have my hydroxyl, my OH group together. This oxygen, super electronegative, wants all of the electrons, super greedy. So it's going to pull in this hydrogen and it's going to pull this electron off this hydrogen. And this is basically just a hydrogen proton, that's it. But the electron is here around this oxygen. This hydrogen is now very positive. It doesn't have any electrons. So this positive charge is going to pull other water molecules towards it. So here I have the molecule. I have that positive pull of that hydrogen, and a water molecule is going to come in. This end of the water molecule is very electronegative because of the oxygen. This part here is very positive. This electronegative is going to be attracted to this hydrogen. It's going to want to try to pull electrons off. It can't, because remember, this is full. It can't take any more. So it's going to build a bond. What type of bond is it going to form? Remember what the dashed lines mean? That means it's a type of hydrogen bond. So this is like magnets. Opposites attract, it's going to pull it together. Next, talk about carbonyl. Carbonyl are also polar. So here I have an R group. This is going to be my carbon molecule, whatever, set up. But then I'm going to have a group that is going to be an oxygen double bonded to a carbon. I can have them in the middle, which are ketones. I can have them on the end, which are aldehydes, which is something different. But this is still a carbonyl type of group. So here I have my carbonyl. This here represents an R group. I can just have a carbon, something over here. This is another R group, a carbon attached to something else over here. So this is what R groups kind of represent. My carbonyl is here in the middle. So there's my carbonyl. Without oxygen, it's going to be nonpolar. This oxygen, super electronegative, greedy, is going to try to pull other water molecules in, and so it's going to dissolve in water. I can see it written out like this, where I show being attached to something else in my R groups, carbon, whatever, carbon double bonded to oxygen. Here's a carboxyl. Make sure you know the difference between a carbonyl and a carboxyl. A carboxyl is going to have a carbonyl, and what do we call this one again? This is a hydroxyl. So this has both. That's why it's called carboxyl. It's a mix between the two. This one here is considered acidic. So here I have my R group. So this is my carbon molecule, whatever. This is going to have these molecules here on the end. So my hydroxyl, carbonyl, and so here is my molecule. So this part here is my R group. This part right here. Carbonyl is here. My hydroxyl group is here. So I'm going to have that section right there. So if you guys don't remember, pH is all about hydrogen protons. And if you guys need to watch a video, I've got a video. I'll put it in the description below. But you guys can watch a video on me talking about pH and what all that means. So acids increase the number of hydrogen protons. When we have an acidic solution, it's just hydrogen protons. There is something electronegative, oxygen, whatever. It's pulling the electrons off and it's leaving the hydrogen protons behind. So here I have hydrogen. Remember, hydrogen is one proton, one electron. I bring in oxygen. Oxygen, very electronegative, super greedy for electrons. And here I have 6 valence electrons. Oxygen's not happy. It wants 8. So it needs to bring in 2 hydrogens to help it out. So a hydrogen is going to come in and another hydrogen is going to come in. This oxygen is going to try to pull those electrons towards it, and it's going to bring in those electrons. what's going to happen is oxygen is going to pop off these hydrogen protons, but it's going to keep the electrons. So now just the hydrogen protons are in the solution, and that makes a solution acidic. The more hydrogen protons I have, the more acidic, the closer to 1 or 0 I get. The less acidic, the less hydrogen protons I have in a solution, and the closer I get to 7, which is neutral. If I'm becoming more basic, I'm going to have more hydroxyls. I'm going to have less hydrogen ions. And so all of that is what makes a solution acidic. So now I'm going to have a charge of oxygen. Hydrogen protons are going to be floating around in my solution. So it's going to be what we say ionizes to release hydrogen protons. An ion is a different number of electrons. So oxygen has pulled those electrons, it's been ionized, and it's going to release these hydrogen protons, which changes my pH. So if it's ionized, I can show it like this. So I have an O negative, my hydrogens have been popped off, so I'm going to show this oxygen has that electron attached to it. If it's non-ionized, I'm going to show that hydrogen is still there. That hydrogen proton is still attached to that hydroxyl. I can also see it written out similar to this. I have that H is gone. I'm showing that electron is staying, that H is gone. If it's non-ionized, I'm going to show the H here. Methyl is nonpolar, so we've talked about what makes something polar, and polar is going to be something electronegative that is oxygen, or something's going to be trying to pull other things to it. Nonpolar, remember the electrons share evenly in a covalent bond. So here I have my R group. This is going to be attached to some type of carbon molecule. And my methyl is going to be a carbon with 3 hydrogens attached. When I look at my electronegativity chart, these are evenly charged. So they're going to be shared evenly, going back and forth to all the hydrogens and that one carbon. Here's my R group. Here's my methyl group. So these are going to pair back and forth and go. So oxygen, water is not going to want to mix with this side of my molecule. So carbon and hydrogen, similar electronegativity, so it's a nonpolar covalent bond, so it will not mix with water. I can see it like this, carbon, hydrogen 3. I can also see it on the other side. If I have my methyl group is over here on this side, I need to show that this carbon is attached to my R group over here. It's not the hydrogen attached to my R group. The carbon's attached to the R group, and then the hydrogen's attached to the carbon. Here I have my R group, carbon's attached to the R group, hydrogen's attached to the carbon. So you have to show it, it will mirror it in the other way. And this is going to be the only nonpolar one we discuss with AP Bio. Phosphate, these are super important. Remember, if it's considered acidic, that means I'm losing hydrogen protons. If it's polar, I'm going to have some type of oxygen there that's going to be pulling in water molecules. And if it's negatively charged, I'm going to have negative electrons attached to a molecule. So just seeing this, you can kind of start to predict what you're about to see. So here's my R group. This is going to be attached to some type of molecule. And then this is going to be kind of off on the end. I'm going to have an oxygen attached to my R group, and then my phosphate, and then the phosphate will have these 3 oxygens, 2 of them will have hydrogens. Here's my molecule, here's my R group, oxygens attached to my R group, phosphate in here. But now look at this, all right? So here I have my oxygen, and I have negatives here. So what's happened? So those hydrogen protons have popped off, and now I have these negative electrons. I'm probably going to be having some type of acidic solution. I'm going to have hydrogen protons floating in this solution, which is going to more than likely have an acidic property. So my phosphate group is there. And we're going to see these a lot when we talk about like ATP, DNA, RNA. When we talk about ATP, adenine triphosphate. I have 3 phosphates, and these are together. And when we talk about ATP, we're going to talk about it phosphorylating or dephosphorylate. And this is talking about phosphates coming in and coming off a molecule. I can gain energy and I can lose energy based on these phosphates. Phosphates, really important. We talk about those a lot. If I see it ionized, I can see it here. I have a 2-. This is telling me that my 2 hydrogens have left. So I'm going to be ionized. Last one is amino. I believe this is the last one I think it is. Okay, your amino group is going to be charged and basic. So if it's basic, it's going to be pulling in hydrogens. I'm taking hydrogens out of my solution, hydrogen protons. I'm taking them out of my solution to make it basic. I don't want to have it be an acid anymore. So here's my R group. Here's my amino group. I have nitrogen, hydrogen, hydrogen. And here's my R group. Here's my amino group. And N is electronegative. It has a negative charge. So I have all of these hydrogen protons floating around. This positive proton is attracted to this negative charge. So remember, they're like magnets. So this hydrogen is going to get pulled into that nitrogen. And now the proton is going to be attracted. And now I'm going to have a positive charge. Because remember, the hydrogen protons is just a proton. I have no electrons there. So this hydrogen is going to be attracted, and I'm going to have what's called a charged molecule. I'm making my solution more basic because I am pulling in hydrogen protons. I'm changing the acidity of my solution. I'm bringing it closer to 7 or above 7. I am increasing my acidity, my pH. I can show it like this, NH3+. So this shows that it's going to be charged. I have 3 hydrogens, and I have a plus charge because of that proton. I have NH2, which is this version right here. Oh, I lied. This is the last one. All right, sulfhydryl, these are going to be polar. So I'm looking at something that is going to have an electronegative charge. Here I have my R group. This is going to be my molecule. I'm going to have a sulfur and a hydrogen attached. Sometimes it might be an S and an H together. Here is this right here is all that would be my R group. Okay. This part over here is my sulfhydryl group. Sulfur is electronegative. So I'm going to have an electronegative charge. So that means it's going to be pulling stuff to it. What's going to happen is these hydrogens are going to leave. And then I'm going to have a pull between this sulfur and this sulfur. So this makes a true covalent bond. And we'll see this with disulfide bridge. This is called a disulfide bridge. And we see this in hair. People who have really straight hair have very few of these covalent, these disulfide bridges in their hair. People who have really curly hair have a lot. So we talk about this when we talk about genetics and hair. All right, that is it for this unit over covalent or carbon bonds and functional groups. So if this was helpful, I hope it was. Let me know. If not, we'll talk to you guys later. Bye.